Attention to the camera too. I know that.
Okay.
Thank you everybody for coming to Wednesday Night Lab. I'm Tom Zinnan. I work here at the Madison Biotechnology Center. I also work for the Division of Extension, Wisconsin 4-H. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you Allyson Bennet. She's a professor in psychology. She's going to speak with us about animal research, why we need it, and who it helps. And now I get to ask her the 5 questions. Allyson, where were you born?
Milwaukee, Wisconsin.
Like
Should
my
I turn
dad?
this on?
Yeah, that would be good.
Okay.
And where'd you go to high school?
Greendale High School,
And then where'd you go undergrad?
University of Wisconsin, Oshkosh.
You know, for those of us who went to Platteville, we have a few responses for that, but we'll keep going. And what did you study there? What did you study at Oshkosh?
Psychology,
And where'd you go for your advanced degrees?
University of Memphis in Tennessee, also psychology.
And then when did you come here to Madison?
In 2011.
Very good. So it's great to have you here. Thank you very much for coming and speaking on this wonderful night. Would you please join me in welcoming Allyson Bennet to Wednesday Night Lab.
Thank you, Tom. I'm a huge fan of the work that Tom and all of the people in the Biotech Center do on behalf of the Wisconsin Idea and Public Outreach. So we share that in common. And thank you all for coming tonight and those of you who are watching. So the topic I've chosen is a broad topic, and I'll divide it into about 3 parts. So I would like to talk about some of the discoveries accomplished through animal research, and then talk about some of the concepts that shape decision-making. So some of the broad scientific concepts, some of the things we think about both as members of society and as scientists when we're making decisions about animal research. And then I'll end with some of the ethical consideration and focus specifically on UW-Madison and the animal research here. So what that means is I'm going to cover a fair amount of ground, and I will not be an expert in all of it. Hopefully, I'm synthesizing some of the concepts in a way that is helpful. And I'll start out with some disclosures and conflicts of interest that may be apparent, but I want to make sure. So I'm a scientist. I'm a faculty member in psychology. My research is about how environments and experiences get under the skin and affect biobehavioral development, health, and well-being. And this work is with non-human primates, with monkeys. I'm also an educator, so I have the phenomenal privilege of teaching at UW-Madison as recently as 9:30 this morning. And that means I get to work with students who will go on to do amazing things, who are amazing people, and it's really a privilege to contribute to their education. I'm also a citizen. So like all of you, I have a deep interest in decisions that we make that affect each of us, that affect our parents, our children, society, our neighbors, and the world globally. And I'm an advocate for research. So why am I an advocate for research? Well, I think like many of you, you are, after all, at Wednesday night in the lab. I think that research and science are one of the ways that we learn new things about the world. We have discoveries and new understanding that benefit individuals, they benefit humans, they benefit society, they benefit other animals, they benefit the world. It's a way of moving forward and learning more about our world. What I have represented here are different animal research interests at UW-Madison, across our colleges, across species, across settings, from the field to the barns, to the lab, to the veterinary clinic. The UW-Madison Animal Program is quite broad. So it spans our colleges and schools. There are over 900 separate research protocols, so separate lines of research at UW-Madison. There are over 460 separate, what we call principal investigators or lead scientists, somewhere around 3,700 individuals involved in animal research in some way, in support of animal research, in conducting animal research, in learning about it, in contributing to studies. And there are somewhere around 1 million animals in research. I've taken a very broad view here. So these are animals in field studies, lab studies, any kind of research, and about 75% of those are fish. So here we have fish. There are a number of different kinds. One of the most predominant is likely zebrafish. Zebrafish are studied for developmental biology, for toxicology, for many different kinds of research. And there are quite a few of them, as you can see. The next largest proportion of animals in research are mice. That's around 24%. And then there are a range of other species. So cattle, pig, sheep, horse, monkey, gerbil, hamster, guinea pig, dog, cat, amphibians, reptiles, birds, quite a few different species. Scientists study different animals for different reasons. They choose the animal on the basis of the scientific question, the appropriateness of the model. And you can see that there are a diversity of those animals and models. So let's turn to, I've given you just a little bit about the program. What has animal research accomplished? I'm going to give you just a few of many UW-Madison discoveries. We would be here every Wednesday night at the lab for 50 or 100 weeks if we went through every single one of them.
So.
Okay, we're going to need more people for that. So what are a few of them? I'll pose it as questions. So how do we know? How do we know about vitamins and nutritional deficits that cause disease and poor health? How can it be the case that we go in the store and we conveniently have these little labels that tell us in milligrams, micrograms, exactly the composition of a vitamin or a food, vitamin A through whatever? Part of the reason we can do that, some of you may already know this, are the discoveries at UW-Madison, the discovery of vitamin A in 1908 by Elmer McCollum and his assistant, Marguerite Davis. They were co-authors on this paper and this discovery. To do this, they used rats in their research. And in fact, they developed the first rat lab for nutritional science. It was hugely productive and it benefited many people. How? Vitamin A deficiency is the leading preventable cause of blindness. So that discovery was important. It also led to a great deal more nutritional research. Moving to 1923, we have Harry Steenbach, who discovered how ultraviolet irradiation forms vitamin D. That's important in preventing rickets and bone deforming disorders. So it helped make rickets a rarity back in the 20s. That research depended in part on goats. And it didn't end there. So in fact, Hector De Luca was one of Steenbach's collaborators. He went on to use animal studies to discover how the body's organs use vitamin D to maintain steady calcium levels in blood. Patented those discoveries in the 1970s. In the 1980s, deciphered the genetic sequence of the receptor molecule for the vitamin D hormone. He and his collaborators went on to synthesize 800 plus unique vitamin D hormone analogs. Well, how does that benefit anybody? It benefits us because those include pharmaceuticals that impact the lives of millions of people suffering from osteoporosis, vitamin D resistant rickets, bone diseases associated with kidney failure. And in fact, two top drugs, Zemplar and Hectorol, replace the vitamin D hormone a patient's kidneys can no longer make. And by doing so, serves 500,000 people on dialysis. And UW-Madison continues to lead the world in vitamin D research. We can move on to other examples. Where does the world's most widely prescribed blood thinner for heart patients and clot prevention come from? For that, we can look to the 1940s and the work of Carl Paul Link with rabbits, who discovered warfarin, named after the Wisconsin Alumni Research Foundation. That's the basis for Coumadin, which is the most widely prescribed blood thinner in the world today. It's used to treat heart patients and prevent blood clotting. That work involved the use of rabbits. Turning outside of vitamins and medical discoveries or obvious medical discoveries, we can look at other areas of research. So we might ask, how do animals learn and remember? How do we know that, and why does it matter? There, we might look to the work of Harry Harlow and Peggy Harlow at the Harlow Primate Lab. So from the 1930s to the 1970s, we see a body of work in which the Harlows and their collaborative group uncovered ways to test experimentally and rigorously learning, memory, and something called generalization of learning or cognitive ability in monkeys. Those tests, and one of the pictures I've got here is called the Wisconsin General Test Apparatus, are around today. They're delivered via computers or video games, but they're the foundational methods of assessment for learning, memory, and cognition across a wide range of species, from rats to monkeys to humans. You may be more familiar with Harlow's later work on the nature of love and attachment. We could ask, how do retroviruses work? And what we would see is, again, a UW-Madison discovery. So based on research with monkeys, horses, chickens, and mice, the late UW-Madison scientist Howard Timmons shared the 1975 Nobel Prize for predicting and discovering reverse transcriptase, which is a key enzyme in HIV and other retroviruses. That finding underpins much of modern molecular biology and biochemistry, and again, depended on many different animal models. As I said, we could talk about UW-Madison discoveries from animal research all evening and into the next week and month. There are many, many more. They occur across schools, colleges, research areas. They occur across domains that are relevant for human, animal, societal, and environmental health. We could talk about stem cells. We could talk about stem cell replacement for Parkinson's disease. We could talk about ants and how they talk to each other or communicate via pheromones. We could also talk about discoveries from the past and link them to today. And I'll turn to that in a moment. I do want to point out that there's a website here, animalresearchwis.edu. And thanks to the incredible efforts of the research communicators over in University Communications, it's populated with news stories that continually update and talk about the discoveries from animal research here at UW. Of course, UW is not the only place in the world, and animal research and animal research discoveries do occur across time and across place. And that's a really important thing for us. So scientific discoveries, knowledge, medical advances occur across the world. They're shared across the world. Science often has no borders, and it's often the case that our aim is explicitly to share the benefits of research. And we can see that historically and currently. We can see that with the polio vaccine, with insulin, with the cure for rinderpest and agricultural disease, with treatments for HIV, with the COVID vaccine, the Ebola vaccine. So these discoveries and the basic knowledge are shared around the world. The beneficiaries then are global. Now, you might be thinking, some of the examples I used were a very long time ago. Depending on how old you are, it may seem like yesterday for me on some of them. I promise you, as someone in the classroom, they seem like a long, long time ago to others. And they might prompt the question, well, come on, is animal research still needed? Like, we have computers now. So I want to address that right up front. And I want to say that the thing to remember about science is we use many different methods, sometimes within the same lab, within the same study. So it is often the case that animal studies are taking place next to cell studies, computer simulations, and other methods. Scientists use many methods and models to move scientific understanding forward. They also use alternatives where they are available. And they provide the basic information and discoveries that allow alternatives to be developed. So you can't develop an alternative unless you know something about the system. That's how it goes hand in hand. Sometimes people might ask, well, couldn't you just use a computer simulation or an organ on a chip? The problem here is if you don't already know the system, you can't simulate it. So to build a simulation requires knowledge of what you're simulating. With respect to organs on a chip, they can be useful for some things, but not for simulating the entire body or the interaction between systems, which is something we often want to know about. So in vitro methods in cell culture cannot replace the whole system for many questions. Others will say, why don't we turn to human studies? Human studies in general don't provide us with the level of experimental control or random assignment that we need in order to make causal claims. So the answer on the human question is human studies do accompany non-human animal studies oftentimes, but they are not a replacement for those times that we need molecular analysis, tissue, neural recording, or other invasive techniques that are necessary for some questions to support some causal claims. One way I like to think about this is to look at the brain. That's clearly a pretty complicated organ, right? What you see on the left is an MRI image, a very fancy one. MRI was developed in 1971, so a long time ago or fairly recently, depending on how you look at it. But we still don't know a great deal about the brain. And that tan blob of tissue isn't going to tell us without a lot of work. Sometimes people think about it this way. Imagine you lose your keys in your house. Okay, has anyone done that? I've done that. Somebody gives you a blueprint of your house. Will it help you find your keys? No. So knowing the structure of something won't necessarily answer your specific question. So let's take a look at why that matters, why we want to know how the brain works. I'm going to give you some headlines from the last couple of years, things that have received popular press. So artificial spinal cord wirelessly restores walking in paralyzed monkeys. That's pretty amazing. So the target here isn't an intervention for monkeys. This is the work that comes before it goes to humans. And it does go to humans. So we're now at a point where we have headlines like Once paralyzed, 3 men take steps again with spinal implants. So we have what are called neural prosthetic devices that can have the brain talk to the limbs to guide motion. And it's not just motor function. So here we have a headline: neural-enabled prosthetic hand helps amputees feel again. So restoring sensation to people who have lost sensation. This is based in non-human primate research at Arizona State University in Pittsburgh, and it's pretty amazing. We could turn to other clinical disorders, things like Parkinson's disease, where we have new technologies, new treatments that involve deep brain stimulation to reduce the progression of tremor, and stem cell therapies, gene therapies that are holding promise for treating people with Parkinson's disease. So again, things that involve understanding how the brain works. We have advances in spinal cord injury treatment. So here you're seeing work from the Veterans Administration that is designed to treat veterans who have a spinal cord injury and are no longer able to cough, which puts them at risk for frequent pneumonia and hospitalization. Through work with dogs at the VA, that has been addressed. And of course, we have the COVID vaccine. So the COVID vaccine is an example that we'll use tonight to highlight some of the role of basic research and the timescales. So why it is that vaccine could be developed so quickly. Don't worry, I am not going to read this, okay? But if you want to read it, this is a phenomenal article by a colleague, Justin Borlick, at Speaking of Research. It's the history, like why we have mRNA vaccines. What you can see is it goes all the way back to 1971 with frogs and rabbits, and forward to the 1990s, the early days of RNA and vaccines, the 2000s, optimizing mRNA vaccines, the 2010s, vaccine optimization, clinical trials, and of course, what we know today. A long period of time, many, many studies, many basic advances that led to that clinical application, and many different animals. So what I've showed you is a mix of treatments after something has gone wrong and efforts to prevent something from going wrong in the first place. So a vaccine versus a treatment for spinal cord injury. Our goal, I think, and I think a shared goal, is prevention. We don't want to have a treatment. We want the thing to not happen in the first place. So sometimes people will say, but we haven't cured diseases. Are we making progress? Are we? And I'll give you a couple of examples that I think illustrate the fact that we are broadly as a scientific and society. So here's HIV. HIV is not cured, but what I'm showing you is a graph that illustrates from 1981 to 2007, in blue line, the death, in red, living with HIV. And you can see those lines cross. So the number of deaths go down, the number of people living with HIV goes up. What happened? The introduction of effective antiretroviral treatments. So in the 1980s, the average survival with an HIV diagnosis was about a year. That has changed radically. So continual evolutions in treatment mean that many people can live normally with a diagnosis of HIV. We can say something similar for cancer. So people will say cancer isn't cured, and it's true. All cancer isn't cured, but taking the words from Vincent DeVita, who is a former director of the National Cancer Institute, we could say this: some cancers are cured, treatments are improved. So if you can think back to the treatments that predominated in the 1940s, 50s, 60s, 70s, treatments now are better. People have better quality of life. They survive longer. those are advances. We can say that childhood leukemia is now almost completely curable, Hodgkin's disease, almost completely curable. Mortality from colon cancer, breast cancer, prostate cancer continue to drop. Not cured, treatments better, longevity better. So those are a few of the many advances that benefit humans and other animals in the environment. And I'll talk more about those last two in a moment. What's also true is it's a fact that animal research and animal testing played essential roles in those advances and many, many more. It's also true that we're not just thinking about the scientific research and benefits to humans. Rather, what's happening is a balance between scientific goals, new knowledge and discoveries, balanced with compassion and commitment to our moral obligation for the humane care and treatment of research animals. It's also true that the use of animals in research can be an emotional, sometimes controversial issue. It is most certainly a complex issue. And to me, what that means is it merits a special public dialogue based in factual and accurate information, because at the end of the day, scientists aren't making all the decisions. We as a community and society are making the decisions. And so what we need is a dialogue that starts out with accurate information, with an understanding of the basic concepts that are driving decision-making and the likely consequences or outcomes of any particular set of decisions. So what I'll turn to now is a little bit about that. So how do we get a handle on that complexity without getting lost in it? So what are the big concepts? I'll talk about each of these. So one set of big concepts is weighing benefit and harm and the consequences of decisions. So the consequences of decisions might mean harm for somebody, benefit for someone else. One thing I want to note is when we talk about harm here, we're not only talking about what happens if you do a research project, but also what happens if you do not. That's called the harm of inaction. And the basic idea is if we decide not to try and have a vaccine, we won't have a vaccine. That would be the harm of inaction. So it's kind of a big example. Another concept that's super relevant here is the idea of basic knowledge as a foundation for application. And I'll give several examples of that. The idea that from basic discovery to application has a long time scale, and the fact that the outcome and range of impact of a particular study is simply not known in advance. So the reason you ask a question usually is you don't know the answer. The same thing applies to science. So we don't know the outcome in advance of doing the study. And if we fail to understand all of that, fail to understand the nature of science and apply that to our careful consideration and decision-making, I think we're at risk. We're at risk of failing public interest in scientific and medical progress. So let's take a look at those concepts. So how do we develop new scientific understanding, new treatment, prevention, intervention, and improvement? How do we get from A to B? Oftentimes, when people think about this in the context of medical research, they think about what's called the biopharmaceutical research and development process. I'm showing you a cartoon here. There's a little tiny orange area on the far side that says basic research. I'm about to tell you basic research is more than a tiny sliver of this process. And then we see drug discovery, preclinical, clinical trials, FDA review, and approval. Across that, what you see is a very large number of potential new medicines getting winnowed down. More and more are getting lost by the wayside as this process unfolds. Some of those are medications that could be harmful, and we're glad they're not in that pipeline. At the end, what you see is that one approved drug. And sometimes people look at that and say, well, that's super inefficient. That's not great. I think probably biopharmaceutical companies would be very happy to have an alternative that's effective and works better. And I suspect many are working towards that goal. I know that's true. What I would also say, though, is that one approved medication that's effective and safe a bad outcome? If it's the medication you or a loved one needs, if it's a medication that has huge impact for humans and other animals, it's a good outcome. One is better than none. So let's look at those basic discoveries. This is the way I think about it. So when we talk about new treatments, we're up at that little pinnacle. If you thought about this like an iceberg, that's probably how basic scientists often feel. The rest is unseen, but it's that huge set of basic discoveries, understanding how a process, biology, behavior, and immune function works, what's typical, what's atypical, what disease looks like, what systems are involved, how those systems work, that tells you what the targets for treatment are. So basic research is the support for what comes next. This is how I would look at that biopharmaceutical pipeline. I'd put that big basic research right in front of it. They go together. We need both. And basic research is that which is often publicly, federally funded by organizations like the National Institutes of Health, which is you, that's everybody, that's public funding. So what is the value? Why is that the case? Why do we have the NIH? Why do we have federally supported research? Well, because it's important for Americans' health, global health, really. It's necessary to inform policies that are meant to protect public health, and it's irreplaceable. So it's unlikely to be replaced by private industry. And you can think about that in terms of things like air pollution, lead in paint, pollutants in water. There are a lot of examples we could think about. Private industry is not likely to do that work. It's a public question, and we have publicly funded research. We can see that the NIH traces its roots to 1887 in acknowledgement of the value of scientific research that is publicly funded. And I haven't updated this slide in a while. At least 153 Nobel Prize winners have received funding from NIH. I know that's a little bit higher this year. And those discoveries have led to advances that benefit us all. Things like the development of MRI. So that's basic research. We'll turn now to the idea of timescales and range of impact. So the timescales question is something like this. How long does it take? So from the time you have the discovery till you have an application, how long? From the time you plant something to the time it grows, how long? Is there a one-to-one relationship? So should we expect that every study leads to a cure? I'll give you several examples that illustrate timescales. And we'll start out with diabetes. It's well known. We know about insulin. We know what kind of effect it has. What was the time scale? The amount of time between the discovery of insulin and the first human patient receiving insulin, that was about 45 years. So in 1879, the discovery that the pancreas produces insulin was made. How? Hypothesis-driven science based on a clinical human observation and scientists removing the pancreas from dogs. The dog developed diabetes symptoms. Fast forward, 1921, again, hypothesis-driven research, insulin from healthy dogs injected into diabetic dogs. Their normal state was restored. What happened next happened quickly. So insulin was extracted from cattle pancreas. Its dose, purity, and safety was tested in rabbits. By 1922, the first human patient received insulin. By 1923, it was widely available. And the scientists involved received the Nobel Prize, 45 years. Sometimes people forget, because diabetes can be treated more readily now, that early on, here you're looking at an early patient before and after 4 months of insulin treatment. Before the discovery of insulin, type 1 diabetes had an expected lifespan of 3 years. We'll go to another example. How do we know what brains do? So many of you have seen this colorful cartoon figure. Look, all the areas of the brain are labeled. They're in different colors. It says what they do. So we're all used to seeing this. But what we also know is the actual brain is not color-coded. It is not labeled. It does not have instructions for which parts talk to each other and what they do. That had to be discovered. And how that was discovered was through the massive application of many scientists, many different studies, many different human and non-human animal research projects. That's why we have a map or an atlas of the rhesus monkey brain, the rat brain. We know about the nervous system of aplysia and many other animals. And you have to know about the brain parts, the structure and the anatomy, as well as the function, the behavior, things like sensation, learning, perception, memory, emotion, and motor function. So this is where you see two things come together. In order to know about function, you need ways to assess something like learning or memory. I talked about that early on with Harlow. We could also talk about operant conditioning. We could talk about mazes. We could talk about all the ways we've learned how to analyze learning, memory, sensation, and perception in humans and other animals. And that allows us to then ask which brain areas are involved in those functions, and with that, get into the color-coded brain. We talked about neuroanatomy, neural function. I also want to talk a little bit about brain chemistry and function. So that's another area that helps us understand timescales, range of impact, and the role of basic research. So here we are in 1921, Otto Lowy discovers chemical signaling through the use of frog experiments, 1921. By 1935, a guy named Vittorio Erspamer discovered a chemical called, it would come to be called serotonin. Again, using frogs in his research. What happened next? Well, over a 35-year time span, serotonin's structure was determined. It was synthesized for research. A scientist named Betty Turon identified serotonin in mammalian brain across many species, which provided the basis for Wisconsin scientist D.W. Woolley to hypothesize or propose a theory. The theory was that psychoses, mental health issues like schizophrenia, depression, were not in fact due to demons, moral failing, or any of the other current hypotheses, but rather, that sounds so silly to us today, but it's because somebody said, no, this could be chemical function or chemical imbalance. This could have a biological root. And therefore, a biological treatment. So what happened next was an era that is called targeted pharmacotherapy design, which led to the development of Thorazine, antidepressants like fluoxetine, Prozac, and widespread prescription of those treatments to treat people who had things like schizophrenia or depression. Two things here. Number one, are those treatments perfect? No, they aren't. So they are better than the alternatives that existed before that. Things like insulin shock therapy, frontal lobotomy, institutionalization. And in 1955, there were over 500,000 people in the US institutionalized in horrible conditions. Well documented in Life magazine over there on the right. Those weren't great treatments. So our current treatments may not be perfect, they are better. But what that also means is that's why scientists continue to advance studies to make better therapies, better treatment, better understanding. So let's wrap some of this up and say, so why is history helpful? What's the value of going back to the 1920s, 1908, looking at the time scales? Part of why it's valuable is post hoc analysis is what we can use as the basis for projecting potential benefits, potential harms, potential outcomes in advance of doing a study. So as scientists and as people involved in making decisions about science, we don't know the answer when we make the decision. We don't know the answer ahead of doing the study. We have to evaluate benefit and risk on the basis of estimated potential outcomes. And we do that not by guessing, but by applying logic and prediction on the basis of existing applicable knowledge, on the basis of what we know about the nature of science and discovery. Another way to say that is a simple cartoon. So if we think about what impact a discovery has had, we have to ask over what time scale. And if we're talking about the range of impact, we have to acknowledge that something like understanding mRNA has many applications in a broad range of impact. What we also need to remember is that it's dynamic. So what I'm illustrating here is who benefited from the discovery of insulin? When are you asking? Because as time goes on, more people benefit. So that number continues to increase. What I'm also illustrating here is the range of interest holders increases. So at this point, insulin is also used in treatment of non-human animals with diabetes. That might not have been predicted in 1923. So animal research and testing are much broader than biomedical research focused on new treatments for humans. So in fact, they are beneficial to other animals. We can take any number of examples here. I'll focus on a couple. And the first one I'll focus on might be a good discussion point a little bit later. So what I'm showing you here is some fairly recent data on global COVID vaccine rates, so across the world. And you can see that in some countries, there is a quite high access to vaccines, quite high level of vaccination. There's a lot of inequity in there, though. And you can see it just by looking at the world map. On this side, what you see is what we're doing in America, which is vaccinating zoo animals. Why? Because the same vaccine technology, the same disease can affect other animals just as it does in humans. And so one of the world's largest animal medicine developers developed a vaccine for other animals. So you see the headline, the San Diego Zoo's great apes became the first to receive an experimental COVID-19 vaccine for animals. Ironically, we have chimpanzee research in the US again, and they are benefiting from a vaccine that was trialed on mink. So you see a mix of human and animal benefits from the research. We could talk about benefits for pets. So there are many pet owners in the US. Pet medicine is a big industry. Much of that is based on animal research and animal testing, as in the insulin and COVID vaccine examples I gave you. that benefits other animals. And you see Scout, one of the dogs treated at the UW vet clinic. I'm pointing out here that these animal medicine companies, just like universities, are using animals in research and testing. We could look at wildlife ecology. We have researchers studying coyotes, they have studying walleye, they're studying foxes. They're examining things like, how does climate change affect where porcupines range? How do changes in the environment affect other animals? And it's Wisconsin, so we will also have to say other animals that benefit from animal research and testing are agricultural animals, cows, pigs, sheep. And that benefits other animals, it benefits humans. Given this work, improves animal production, health, and welfare for animals in the agricultural industry. So a lot of ways that animal research benefits humans, benefits other animals, benefits society. So this last bit of the talk, I want to turn to some of the ethical considerations. I hope I've given a broad view of some of the core concepts that I think are relevant to ethical consideration. When we turn to ethical consideration, what we're asking about really is, how do we decide what the right thing to do is? We decide that at the level of individuals, we decide that at the level of communities, societies, and nations. So in the case of the use of non-human animals by humans, there are a lot of different ways that humans use other animals: food, companionship, entertainment, clothing, labor, education, product testing, research, lots of different ways. For some people, all of that would be unacceptable. For others, all of it would be acceptable. So these are our poles. For most people, we're somewhere in the middle. So that's our fundamental or first question. Is it ever acceptable for humans to use other animals? There are positions and points of view expressed as follows. No, it's never acceptable. So these points of views might be an animal rights point of view. Other animals have rights that are the same as ours. They have the right not to be used. Or a plain absolutist position, which means humans should never use other animals no matter what. These positions are different from an animal welfare position. So I think many of us, most of us, take an animal welfare position. We're concerned about the humane treatment of animals, but we may or may not be opposed to any particular use of those animals. So that fundamental issue, whether humans should use other animals, is point one. The second one is it depends. And it depends on a lot of factors. So whether it's acceptable or not might depend on the purpose. What are you trying to accomplish? On whether there's an alternative. Can you do something else? Whether there's potential for pain or suffering, it might depend on the species. It might depend on the consequences. So all of those likely play a role. In the US, formal ethical justification is required for almost all non-human animal research. We're going to leave these other uses of animals aside. We don't have the same requirement there. But I will make the point that historically and currently, ethical decisions about human and non-human animal research are very tightly intertwined. And we can see that in the US, we can see that in global codes of ethics. I've chosen just a few examples here. So we have the 1906 Pure Food and Drug Act, that's the US. And that's what led to the creation of the FDA that we still have, public federal agency. It requires that before treatments, devices, or drugs are evaluated in humans, they first be evaluated in two types of animals. Similarly, we see in 1948, following World War II and horrific experiments, a code of ethics that would become a worldwide code that specified before human studies must be based on the results of animal experimentation. So prioritizing humans over other animals to protect human rights. We see that in the Declaration of Helsinki, which is a current world medical code of ethics. Research with humans should be based on the results from laboratory and animal experimentation. This is how human and animal research ethics are intertwined. And there are many, many examples that tell you why that is so. I've chosen a couple here, a book by Susan Lederer on the UW-Madison campus, a book by Harriet Washington, and both of them will tell you a lot about the incredibly disturbing history of human experimentation and horrific human rights violations that led to contemporary ethical codes. Those ethical codes are speciesist. So they prioritize humans over non-human animals. They evaluate harms and benefits by considering individuals, species, society, and the environment, but prioritizing humans over other animals. That doesn't mean that the other animals aren't given moral consideration, that humane care and treatment isn't prioritized. It means that our world's ethical codes reflected in Helsinki, so that's not the whole world, and there's certainly a lot of variation, prioritize humans over other animals. So the basic principles regulating animal research or contributing to decisions about animal research across many countries are: one, is it justified by the purpose and necessity? So what's the purpose? What's the necessity? What's the potential harm and benefit? If there's an alternative, use it. If it's justified, then how is animal welfare protected in balance with scientific objectives? Use the fewest number of animals needed. If there's potential for pain and suffering, it should be minimized, as it would be in humans. So where does that ethical consideration occur? It occurs across many different levels. So it's not a single place. And I think that's important to remember. So it occurs in researchers' selection of questions, selection of methods, their experimental design. It occurs at the level of funding agencies. So what NIH does and does not fund has an element of risk-benefit analysis, ethical analysis of the purpose and potential benefit. It occurs at the level of what are called institutional animal care and use committees and public agencies that oversee animal research. So let's turn to those. So now I'm turning to, depending on what you like, either the super boring part or the really interesting part. So if you're into public policy and laws, you're going to get a little bit. So what are the key components of US laws and standards? Let's start out with, again, history going from 1900 to the 2000s. We won't talk about each and every one of these. What I want to convey to you is continual progress and evolution, both in terms of human research participant protection and non-human animal research. Human research is in red, non-human animal research in blue. We go back to the FDA, the Pure Food and Drug Act of 1906 for humans, and the first codes for ethical care of laboratory animals in 1909, 1925, a long time ago. But let's go to the federal laws. So in 1966, we see the Animal Welfare Act preceding. It's a major federal law about the care for non-human animals preceding the 1974 National Research Act that governs the protection of human participants in research. And then in 1985, another animal research law, the Public Health Policy. So let's take a quick look at those. Here we have the Animal Welfare Act. What is it? It's a set of standards. So it's a law that governs the care of animals in captivity for many, many uses. There are about 8,000 facilities, institutions, organizations that are either registered or hold a certificate with the United States Department of Agriculture, which means they're overseen by the USDA to monitor compliance with the Animal Welfare Act. About 14% of those are research facilities. The majority are breeders, dealers, exhibitors, places like zoos. What does the Animal Welfare Act do? It specifies many things about the care for non-human animals, that alternatives are considered if procedures are painful, that proper anesthetics and analgesics are used if there's potential for painful procedures, that institutions have an institutional animal care and use committee. There are rules about exercise for dogs, enrichment for primates. Note here that the act does exclude purpose-bred rats and mice for research, but they are covered by a different law. And that's important because worldwide, the overall estimate is about 96% of warm-blooded vertebrates in research are rats and mice. The majority. I think sometimes, especially if you're around Madison, you might get the idea that it's mostly monkeys. It's not, it's mostly rats and mice. And so it's important that rats, mice, and fish are covered and have federal oversight via a different law. So public health policy, and that's administered by the NIH. The two federal agencies work closely together. We might have gotten too far in the weeds there, but sometimes we hear this idea that rats, mice, and birds aren't covered by laws. They are. So what's it look like? We'll take it to UW. Here's a way you can think about that oversight and think about your role in it as well. So through the democratic process, our elected representatives enact laws. One of those laws is the Animal Welfare Act and supports federal agencies that oversee the use of animals or care of animals in captivity. So that's the USDA and the NIH. And then we have local oversight as well via our institutional animal care and use committees and other sources of authority. We also have voluntary accreditation programs by longstanding groups that come and inspect all of the animal facilities, protocols, and functioning of the program. So this is a range of some of the oversight. At UW-Madison, we have an institutional official as one of our authorities. We have a chief campus veterinarian as a second authority, and we have institutional animal care and use committees. We have an unusual system here in that we have 4 separate ones corresponding to our colleges. And each of those committees by law must have a scientist, a veterinarian, and a public member, someone not employed by UW-Madison. They actually have many more people than that, but that's a minimum. Those are types of expertise that must be represented. Across our 4 committees, 43 voting members are involved, 16 alternates, and those IACUCs review and approve every single proposed animal study before it can begin, and then continue to do reviews. So they do program exhaustive inspections and program reviews. If you're interested in what goes into an IACUC protocol, it's a lot. It's a complete description of the study, the significance, the justification, why you're using the species that you're using. It tells who's going to be doing the work, what their training is, whether they've completed mandatory training. And we have all of this on the website. The people involved in animal care after those studies are improved include veterinarians, vet techs, animal care techs. We have hundreds of those. And they are dedicated, committed people who are taking care of the animals, feeding the animals, observing them, monitoring their health. So again, somewhere around 3,700 people involved in supporting or conducting animal research. And together, all of those individuals, groups, and organizations are responsible for advancing scientific discoveries and humane care of the animals in research at UW-Madison and research around the world. So I'll wrap up by saying one thing about the future. So if you ask, what does the future hold? One of the things we might note is that the future is pretty likely to hold continuing and new challenges to human, animal, and environmental health. This seems very likely. New viruses, new threats to human health, continuing threats via diseases we already have. It's dynamic. Climate change, environmental change will be new threats to human, animal, societal, and environmental health. And what that means to me is that we need to pay attention to our decision-making for a number of reasons. Because as a scientist, I believe that facts, evidence, and science matter, and that how well we address those global challenges will depend in part on scientific research. It is one of the tools we have that is effective, that can help us address challenges in a way that benefits everyone. But scientists don't make all the decisions. That is all of our jobs. And so informed public decisions matter. And those decisions now will affect the capacity of young scientists, future scientists, to be able to make discoveries, advance knowledge, and global health. So the decisions we make now are going to affect what our graduate students, undergraduates, and new generations of scientists can accomplish. So it's for all those reasons, I think it's really important to have very serious conversations based in fact about topics, especially those that can be controversial. And with that, I will stop and ask if anyone has any questions, and thank you for your attention.
I greatly enjoyed your talk, and I guess I'm interested in the 2016 paper about the social responsibility to engage the public. I noticed you have 23 citations. I wonder, in retrospect, and looking at those citations and your talk tonight, what has changed since 2016 and how you engage the public and the attack on scientists that we have today.
Is this on? Yes. Okay, I have to confess to having a little bit of a hearing deficit. So I think what you ask is about a 2016 paper on scientists engaging the public.
Yes.
Okay. And I think you're asking, has anything changed?
Yes.
I think so. So I'm a pretty optimistic person. I'll give you that. But I think many more scientists are very actively engaging in dialogue with communities around the world about scientific research and scientific findings. I know of one example, there's a global neuroethics group that is working with scientists across the world and communities to talk about very complex scientific ethical interplay issues that would include primate research, for example, but also things like neuroprosthetics. So these are advances that are going to be expensive, for example. how will we have equitable distribution? It's also true that people vary in their view of the brain and the appropriateness of implanting something in the brain or messing with consciousness. So that very active engagement, I think, is happening. Is that responsive to your question, though?
Yes, thank you.
Oh, good. I also, one thing, I see Jordana Lennon here, who's the outreach person, brain at the Primate Center, who works with phenomenal numbers of people in Wisconsin and beyond doing outreach. Tom's here. And the next generation of scientists, I think, are more and more inclined to do outreach activities and public engagement. Is that fair?
Jordana, would you like to address Sandy's question? Several decades?
Wow, that's really fun.
Yeah.
Yeah, I'll turn around. I'm Jordana Lennon. I've been the Primate Center's communication and outreach specialist for 25 years as of next month. And I'm gonna just take it back to what Dr. Bennet showed, the Animals in Research and Teaching website. It's a lot of work has gone into that site. And we're trying to be very transparent and with the vast numbers of animals and at UW Madison, and we have about 1,600 monkeys at our primate center. Sometimes we're not perfect, we strive to be, but there have been some violations reported on USDA inspections and some injuries, animal escapes, clogged water pipe. These are reported promptly to the USDA and they are posted on the site. And so we try to be very open about that. Recently, I've always heard from people who are very supportive of the research, just no matter what, and who are completely against the research, no matter what. What I'm hearing from a lot of people that doesn't always get the attention I think is warranted is they call me, they email me, they post on social media. We've served scientists, myself, students, and others have reached in person almost 100,000 people in the state since 2010. And just having a dialogue with someone face to face is so helpful. Probably I would say more than social media. But what I'm hearing now over and over again, and I'd love to have an answer for this, if there is one, is there's that kind of troubled middle that you don't always hear from. People that tell me, call me on the phone from New York or California, I want to support the research, I think it's valuable, but isn't there any way we can put these monkeys into larger cages? And I don't know, I mean, we are one of 7 national primate research centers, and there's 2 of us, I think, that all our animals are housed indoors because of climate and other reasons. But the question I get is, can they be in larger enclosures, much larger, have grass under their feet, just more room to exercise, that sort of thing. And I don't know if it's because it's on the federal taxpayers too, especially like in Wisconsin and Washington State, spend millions, if not billions more dollars. We have to prove that to build these enclosures and outdoor, indoor because of our climate, or is that more difficult because if you have larger closures, it's harder to get around with the monkeys, or are there environmental things that can get in and mess with the research? Do you know what I'm saying?
Yeah, I can take that one if you
Thank
want. So I'm also going to remind myself of something I want to refer to. So Wisconsin is cold in the winter. We all know that, we're here. So we are one of the places where the animals are in fact housed indoors, as Jordan's just said. But I think what's important to remember there is the outdoors and indoors is a balancing act, a harm benefit act, just like everything else. What do I mean? Well, if we have animals indoors when we're trying to study viruses or development, we're controlling the environment. So the purpose of having animals in a captive setting is to conduct the science. If you undermine the science, then you've undermined your ethical justification. So that's the balance. So if your animals are exposed to air pollution, as they are in California, exposed to valley fever, as they are in Arizona, exposed to pathogens that are outside, you've got a potential problem for the research, but you may have a potential gain in that the animals have larger naturalistic environments. So it's really a balancing act. And I think that's part of what I tried to convey tonight is much of this isn't simple answers. And that makes it a little bit hard. What I've also put up here is a proposal for accurate labels for drugs. I forgot to mention this earlier in part because it's a little complicated, but I think part of when people call, when people want to be engaged, part of that is coming from a place where we don't do a great job of communicating how the vaccine depended on 40 years of research, or how a particular treatment depended on loads of basic research, how NIH-funded work contributes to that ultimate benefit. So we have a proposal that drugs be labeled in a way that allows people to see that. Are there other questions?
Here's one from Raj online. Have they studied the impact on drug discovery delay by the Animal Protection Act of 1986 and the increase in human deaths? Question. In this case, humans died and animals thrived. Is this considered a balanced approach? I think what Raj is saying is with this act, he's claiming there's an increase in human deaths because of the more difficult animal research. That's the premise of this question.
I think the premise of the question is, correct me if I'm getting it wrong, as you introduce barriers to scientific research with non-human animals or make it more difficult, are you delaying advances and leading to bad outcomes for humans? Does that sound about right?
Let's say that that's the question, and Raj can tell us by chat if that's right.
This is kind of like being in a classroom during COVID. Everything was in Zoom and it's kind of great. You didn't have to wear shoes. People put their questions in chat, no problem hearing.
It wasn't just the shoes.
There were no shoes. I'm not sure I'm going to have an answer for that question because I don't know how you showed the negative. I was just thinking about that. So we would need evidence that things didn't move forward. And I'm not sure how we would get that. I do know that it has become increasingly difficult there are increasing burdens on research. There are estimates, I think it was the National Academies estimate how much time scientists spend on paperwork. It was like 43% of their time. That's a lot of time. I felt that one. And I may be a little bit wrong, but there's a lot of administrative regulatory burden. That's good in some ways, but it has impact in others, right? I do know there are countries that have banned the use of particular species for research. That has most definitely stymied and impacted their research programs. It has sent their scientists to other countries. And we have high profile examples of that. Non-human primate neuroscientists, for example, moving from Europe to the US or China. So I know there's impact there.
And Raj did send a note saying, yes, that's what I meant. So
Oh, good.
Other questions?
So I had a question regarding the in silico drug development cycle, specifically looking at the question of can you do research with computers and why do we need animals? And in many ways, the best we can do for computers is really based on the model. And the model is dependent on our understanding about complex systems and how they interact or don't interact. And in many ways, the only way to even verify that is through animal studies. So I just would like to say thank you for pointing that out. Is a lot of times as a computer scientist, I get those types of questions saying, "Mike, just do drug discovery in silico." We're called complex systems. And the funny part about complex systems is they fail in complex ways.
Thank you. Yeah, I think you're also illustrating the point, which is you go back and forth. So thank you for pointing that out. It's like, can we verify? We go back, we go forth, and we use many different models. I'd also put in a plug for, all those places that say not tested on animals. That little logo. Oftentimes, the reason that is all of the ingredients have already been tested on animals. My daughter, probably I shouldn't say this, but going in some of those stores is problematic because I always wanted to say, but they were tested on animals, right? Like that's how we know they're safe.
Other question? You mentioned you went to UW Oshkosh, I went to UW Platteville. What are these regulations that Raj was referring to? What has that done to increase or decrease access of undergrads at comprehensive universities like UW Oshkosh, UW Platteville? Can they afford to do that? Do they have the infrastructure for this administrative burden?
That is a perfect question. So the question is, I think I can handle it in a little bit of story form. So I've done a fair amount of service work with the American Psychological Association. It's been around for 130 years. And they have a committee on animal research and ethics, which they've had since 1925. So they did a survey a few years ago. It used to be the case that many psychology departments and small colleges had rat labs or had laboratories where students could learn about animal research, learn about fundamental principles of learning, memory, and develop those skill sets. This was true at the University of Wisconsin-Oshkosh when I was an undergraduate, which was not 100 years ago. It was true then. I don't know if it's still true, but what I do know is that the APA survey showed many departments no longer had animal labs for that reason. It's expensive, the regulatory burden is high. There also may be fewer people trained. So that future scientist part matters, right? Because if nobody's trained in particular techniques and methods, it will go away. So my guess is the impact has been substantial. I'm feeling like I need to go check that out about UW Oshkosh right now, and I hope I haven't said the wrong thing. But I do know the reason that I went on to get a PhD in psychology was my undergraduate research experience. That was phenomenally important to me. It was working with rats. I in turn train undergraduates in my laboratory here. This is one of the things I think is vitally important. It's the next generation of scientists. And I said that at the beginning, we have phenomenal students. It's really a privilege. But so does UW Oshkosh and Platteville and Eau Claire and the others.
Any other questions?
In privately funded research, what provisions are there for animal welfare, if any?
That's a great question. And the answer to that, this is going to sound like a refrain. It depends. So private industry in the US falls under the same Animal Welfare Act that UW-Madison publicly funded universities fall under. So does Zoetis, the big animal medicine company, but it depends on the type of animal. So the Animal Welfare Act covers dogs, cats, monkeys, rabbits, gerbils, hamsters. I'm sure I'll forget some. It does not cover laboratory mice, rats, and birds bred specifically for laboratory research. So for public universities, we fall under, or publicly funded research, we fall under this other law I talked about, and OLA oversight. Our IACUs look at all the animals, all the animal research, and they report those numbers. They have oversight by OLA and ALAC. For a private company, they won't fall under that rule. So the mouse and rat work won't fall under the Animal Welfare Act. It won't fall under OLA. I'm not in private industry, so what I do know is sometimes there's FDA oversight, and the major thing to remember is that voluntary accreditation. So that's where voluntary accreditation agencies that provide external oversight, accreditation, inspection, and so forth become really important, I think. So that's their way of assuring that they're following the laboratory animal guide and all the rules and care for animals. Does that answer? There's probably much more than you wanted to know. So a lot of people are pretty actively debating, like, well, should the Animal Welfare Act be revised so that it in fact encompasses all of those animals? The question is, there are 8,000 institutions that the USDA is overseeing. How many will we add? And what will cover that cost and so forth?
Other questions? If not, thank you very much. For this year's pharmacy here in Wisconsin. Thanks for coming tonight.
Thank you very much.